Hello and welcome to this flipped classroom edition of biology. Today we're going to be talking about structure and function. Now the main reason I've decided to do a flip of this lesson is because, to be honest, most of it is pull your teeth out boring. It's basic content. That doesn't mean it's not important. It's mostly structural stuff. It's mostly memory based stuff, not a lot of hard concepts in here. It is stuff you need to know, and it's definitely stuff that you'll be tested on, particularly next lesson, but a lot of you would have done some of it before, so it means that you can go at your own pace if you would like to. Make sure you take notes though, and make sure you draw all relevant pictures, as I said, because although it is a little bit boring, it is very, very important that you understand the basics of cells so that you can move forward properly. So we'll start out by looking at cell theory. Cell theory, of course, is the theory which governs cells. We've talked a little bit about this in previous classes. There are three parts to the cell theory. The first one is that all living things are made up of cells. So if you are a living thing, you have to be made up of cells. Everything that we've ever found that is living has consisted of at least one cell. Number two, cells are the smallest working unit of all living things. Now there are smaller things inside organisms, of course, and we'll talk about that in a minute, but not things that do work. Number three, all cells come from pre-existing cells during cell division. Apart from, of course, the original cell, where we don't know exactly how that worked, all cells don't come from base elements, they all come from pre-existing cells during cell division, things like mitosis or meiosis, which we'll talk about later on. Now, if you're looking for a standard definition of a cell, which you can see over here, what we would say is that a cell is the smallest unit that is capable of performing life functions. Now, like I said, there are smaller things inside cells. There are elements and atoms and that sort of stuff, but they are the smallest unit that is capable of performing life functions. Those are life functions that we talked about a few lessons ago. So they can move, they excrete, they respire, they grow, they die off, things like that. Now, there are two main types of cells that we're going to be talking about today, and we're going to be focusing specifically on one. The first one that we're going to be focusing on is eukaryotic cells, which you can see here, and the second one is prokaryotic cells. Now, we're just going to have a basic look at these two at the moment. Prokaryotic cells in particular we're going to focus on later on when we look at microorganisms. Eukaryotic cells we're going to delve a little bit deeper today. But for the moment, what I want to do is just look at the base differences between them. Now you can see I've done an amazing drawing. This wasn't Picasso, this was Corey Barrett original, of a basic eukaryotic cell here with a few what we call organelles, and we have a basic prokaryotic cell over here. Now the main difference between the two, eukaryotic cells are used by multicellular organisms. Okay, so they are, so we'll see down here, multicellular organisms. So things like plants and animals. If you are big enough to be seen, basically, you must be made up of more than one cell. If you're made up of more than one cell, then you must use eukaryotic cells as your basic cell type. You also must consist of membrane-bound organelles. So eukaryotic cells have membrane-bound organelles inside them, and that means that they have little organelles like we can see. I'll just highlight for you here and here. Basic structures that are surrounded by membranes. Thirdly, they are much, much larger than prokaryotic cells. Okay, we're looking at sort of 10 to 100 micrometers in size. Prokaryotic cells, on the other hand, main differences, they're used by unicellular organisms, so things like bacteria. So if you're an organism that is one cell only, then generally you will always be a prokaryotic type cell. Okay, they have no membrane-bound organelles, which means they have no nucleus. Okay. There are lots of organelles, or little organs, inside of eukaryotic cells we're going to talk about today. Prokaryotic cells have none of those. The main one, though, that you can get tripped up on is nucleus. For some reason, everyone thinks that everything has a nucleus. That's not the case. Everything has DNA, and we can see here, this squiggly bit here is supposed to represent DNA, but that DNA inside this prokaryotic cell is just found floating around within the cell. Whereas, of course, a eukaryotic cell, I'm sure you'd remember from basic biology, is found inside the nucleus, which we'll talk about later. But they have no membrane-bound organelles, and as I said, no nucleus. They're also much, much smaller. Okay, we're looking on the order of 2 to 10 micrometers in size, and that size difference has a lot to do with the fact that they're just much simpler. Okay, so they're the main differences between the two types, eukaryotic and prokaryotic. It's really important that you remember that there are two different types, but for now we're going to move on and focus on eukaryotic cells because we've got a lot to get through. Prokaryotic cells we'll come back and we'll look at in a little bit more detail when we do microorganisms. So let's have a look. Two different types of eukaryotic cells. The first ones are animal cells, and the second ones we're going to look at will be plant cells. We'll start with animal cells because we are animals, so they seem a bit more interesting, I suppose. What we're going to do is you'll see there's a space on the left-hand side. I'm going to draw an animal cell in. What I'd like you to do is draw that along with me. Of course, your drawing won't be as good as mine, but that's to be expected. But have a go, so get some paper out while I'm blibbering on. Make sure you've got it out ready. Of course, you can press pause, I suppose. And then when you start back up, we'll have a go at drawing a cell together. There are a number of different organelles you can see on the right-hand side. We're going to go through each of these. Your main job from today, I suppose, apart from knowing cell theory and the basics between prokaryotic and eukaryotic cells, is going to be to know each of these organelles, their structure and their function, and what they look like, so be able to draw it. And we're going to try that today with animal cells, and then we're going to move on and do it with plant cells. So I hope you've all got some paper handy. Let's have a go at drawing an animal cell. What I want you to draw is basically a big circle. So I'll do here in the middle of the page. Make it fairly big, make it about half a page, because you're going to be putting a bunch of stuff in it. Now that circle, of course, that black line that we've just drawn, is supposed to represent the cell membrane, which is the first of our organelles here. So I'm going to write cell membrane on here. Okay, it's very, very important to label our diagram, so make sure you're doing that, please, as you go along. This is our cell membrane. The cell membrane is not a wall, it's not a structure, it doesn't really have a structural purpose. Don't think of a cell membrane as like the walls of your house or the walls of your room that you're in right now. Think of it more like a Chuck's cloth, okay? Floppy, things can go through it, like water can go through it, but other things can't. It's definitely a structure, but it's much floppier, okay? So a cell membrane is an outer membrane of a cell that controls movement in and out of a cell, okay? You can see that on the right hand side here. Its job is to control movement in and out. It doesn't have a structural purpose. It can consist of a double layer in some cells. In this case though, it also does. It's got a phospholipid bilayer, so a double layer of cells, which we'll talk about later on next week when we look at cell membrane in much more detail. We are going to focus in on the structure of a cell membrane. For now, I just want you to draw it as this circle. Like I said, next week when we look at movement through membranes, we'll be going through that in much, much more detail. Now, the next organelle on our list here is nucleus. A nucleus's job is to direct cell activities because it contains genetic material. It contains DNA. DNA, or deoxyribonucleic acid, is your genetic material. It contains information for making proteins. It contains information for making you, whether you've got blonde hair or blue eyes, or you're tall or you're short, or you're a boy or a girl, or whatever. All that information is found in your DNA, which is stored inside of your nucleus. It's separated from the cytoplasm, which we'll talk about in a minute, by a nuclear membrane, okay? All membrane-bound organelles have a membrane. The nuclear membrane is a particularly important one because it has holes in it, and we'll focus on that a little bit later on in the course. So let's have a go at drawing one in. I just want you to get if you've got different colours, that would be great. We're just going to draw the nucleus as a circle there, simple as that. And just to remind you that it's got DNA in it, we might just draw, it's not very Picasso-esque, some DNA like that. Okay, so that's our nucleus. And again, we will label it, very important to label. Now our next organelle on our list here is the cytoplasm. I'm not going to label that or draw that on your diagram here because the cytoplasm is just the liquid in which everything floats. It's basically the liquid inside of your cell. It's a gel or jelly-like mixture surrounded by the cell membrane, of course. And as I said, all the organelles float around inside this cytoplasm. We're going to use that term a lot because anytime we're talking about anything that's inside a cell that isn't actually inside of a organelle, we're going to say it's in the cytoplasm. Now our fourth organelle here is our endoplasmic reticulum. We'll have a go at drawing that one on there. Funny name, that one, endoplasmic reticulum. Now we're going to draw a couple of them. We're going to draw one here We're just going to draw 4 curvy lines like that, and we might draw one out here, and I'll explain why in just a second. Now you'll see down here with our text that endoplasmic reticulum, I've shortened that to ER, and I'll allow you to use that in a test. I do expect you to write endoplasmic reticulum to start off with, and then you can shorten it to ER and use it from then on. Now you'll see here that there are 2 different little codes here. There's SER and RER, and that's because there's 2 different types of endoplasmic reticulum. Regardless of the type though, they both do the same basic job. Their job is to move material around the cell. Think of them as your local courier, like your little poster. You can pick up posts from the post office and bring it to your house, okay, or vice versa. The endoplasmic reticulum's job is just to move material inside of the cell. It might move it from, say, around about the nucleus over to an organelle somewhere else, or it might move it over to the cell membrane. Now there are 2 different types. There's what's called SER, or smooth ER. Smooth ER lack ribosomes. And then there's rough ER. Rough ER contains ribosomes embedded on their surface. So we'll have a go at drawing some of those in. So this one here, the one that surrounds the nucleus is nearly always the rough ER, and you'll learn why later on when we look at something called protein synthesis. But for now, know that rough ER is rough because it contains ribosomes. If you ever draw one, make sure you draw it with lots of dots on it, because those dots are going to represent the ribosomes. And we'll talk about ribosomes in just a second. But the basic difference is, if you're talking about rough ER, which we are here, so we'll just label this here, RER, the rough endoplasmic reticulum's job is to package and move proteins. Whereas the smooth ER which we'll put down here, its job is to package other things. Okay, anything else. It could be waste products, or it could be things like cholesterol or carbohydrates or stuff like that. Okay, so 2 different types of organelles there. Their job though is to package and move things. Okay. And next organelle here is our ribosomes. Now, ribosomes are very, very small. You can see them. I've already drawn some over here with our endoplasmic reticulum, like little black dots. We'll draw a few more in because they're actually found all throughout the cell. So we'll draw some over here, not just on endoplasmic reticulums. Their job is to make proteins. That's what they do. They do a process called protein synthesis that we're going to learn later on. Where they're going to read some information from our DNA and they're going to make us proteins. Okay. The main reason they're found floating on the ER, of course, and now I'm sure you can recognise, is because the job of rough ER is to package and move proteins. So the ribosomes make the proteins, the endoplasmic reticulum packages and transports those proteins. We'll just have a go at labelling there. So make sure you've got some dots in there. We won't label this one here because it gets confusing. We'll just get some of these dots we've drawn out here and we'll label them as ribosomes. Now, our next organelle on our list is a mitochondria. We'll have a go at drawing a mitochondria in. Mitochondria have a football type shape, although they don't always have to, depending on how the cell has been cut when it's being made. But for now, we'll draw it as a football type shape. So, this sort of idea. Now, this one has a double membrane. So, we're going to draw a squiggly curvy line like this on the inside. This is what it looks like. Okay, and we're going to label that again as mitochondria. There it is. So the job of mitochondria is to release energy through chemical reactions. This is a process called respiration that again we're going to learn a little bit later on in the course. But for now I want you to know that the mitochondria is the site of respiration. It's the site of the release of energy. So all the energy that your cell needs, it gets from the mitochondria. You may have heard the mitochondria is the power plant or powerhouse of the cell. Most students would know that. That's very basic Year 8, Year 9 type knowledge. What I want you to now say is that the mitochondria is the site of respiration or the release of energy through chemical reactions. It's quite a multi-use organelle. It can also do things like recycle and decompose proteins, fats and other carbohydrates. It's good at breaking stuff down. Okay, part that process of breaking down allows for it to release energy. Now our next organelle in our list here, we're getting towards the end of the animal section, is a Golgi body. Okay, Golgi body looks like this, and no points for guessing which one it looks similar to. We'll draw one down here. I can find my cursor. So we're going to draw two lines that curve like that. Then we're going to have a go at drawing a straightish one. Then we're going to draw two more that look like that. Okay, oh, that was not so great. Let's try again. I can get my cursor. There he is. Like that. Okay, so two that curve out this way, straight one, and two that curve out that way. We're also going to draw a few little circles around the outside of them. We can draw circles to represent some little packages which we'll talk about in a minute. Now this is called a Golgi body. Now Golgi body looks a lot like a rough or a smooth endoplasmic reticulum, and that's because they have a very, very similar job. They package and move material. The big difference with a Golgi body from an endoplasmic reticulum is that you can think of a Golgi body as an international courier, whereas like I said before, your endoplasmic reticulum is like a local courier. Generally what you find is that the endoplasmic reticulum packages molecules, sends it to the Golgi body repackages it, and then sends it outside of the cell. So it sends things to the membrane and then out of the membrane to the outside of the cell. Still inside your body, but just outside your cell. Lots of cells will do this. If your cells are trying to release hormones into your bloodstream to other parts of your body, they might make those proteins, package them at the rough ER, send them to your Golgi body. They might get repackaged at the Golgi body, sent to the cell membrane, and then released from your cell membrane into your bloodstream. So Golgi body, really, really important job in packaging materials for external transport. Okay. We've got two more organelles here on our list, getting towards the end. Centrosomes. Centrosomes are organelles which are only found in animal cells. They have a sort of cylindrical type shape to them. So we're going to have a go again at drawing some. So they look sort of like a cylinder like this, with another cylinder on the side like that. Okay. Centrosome's job is to produce what's called spindle fibers. Spindle fibers are like big, long protein ropes, and these things are going to really, really help in cell division. So when we talk about mitosis in a few weeks' time, we're going to talk about spindle fibers pulling DNA around. Okay, so that's what centrosomes do. And there they are. The last one is vacuoles. Now we've already drawn some vacuoles. You probably wouldn't know where, but I'm going to tell you now. These small circles which we drew before, these are vacuoles. Vacuoles are produced at the Golgi body. They're just membrane-bound sacs for storage. Okay, storage, digestion, or waste removal. Think of them like a backpack. Open it up, shove some crap in there, zip it up, transport it around. Okay, that's what a vacuole is. If you want to move things around the cell or move things out of the cell, generally you'll package them into a vacuole, just a membrane-bound sac, and then you can move them around. So we're going to draw just a smaller one here off to the side of our Golgi body, and we're going to label that vacuole. And there it is. So this is our basic animal cell. I hope your drawing is somewhere near as good as mine. Make sure you know what all those organelles look like. Make sure you understand their structure and their function. Okay, really, really important. Now we're going to move on and we're going to look at plant cells. So it's not going to take us as long because plant cells are eukaryotic cells, just like animal cells. So they have very, very similar structures. In actual fact, they're the exact same as animal cells that you saw above, except for a few things that plant cells have that are extra to what animal cells did have. So I've already drawn a basic cell for you here. You can see that cell membrane on the outside. You can see our ribosomes, rough ER, smooth ER, nucleus with our DNA on the inside, and our Golgi body down here with our vesicles. Okay. However, plant cells are a little bit more complex again, because they have to be. They have three things that animal cells don't have, and I expect you to know the differences between plant and animal cells. Plant cells, first of all, have a large permanent vacuole. Okay, vacuole and vesicle, those words are interchangeable for our purposes. They both just mean sacs for storing things. But in this case, plant cells have a large permanent one. So I'm going to draw one in blue. And again, you should be drawing along. So pause the video if you need to and draw yourself a big sort of rectangular shape with a membrane. Draw the nucleus, rough ER, smooth ER, ribosomes and Golgi body. And then probably get a different colour if you can and draw a big large oval in the middle, and that becomes your vacuole. So we'll have a go at writing that in. Okay, that's our vacuole there. Now, the vacuole's job is to store water and maintain plant cell structure, okay, so plant cell shape. Plants can pump water into that vacuole. That vacuole gets nice and tight, and what we call turgid. That puts pressure on the outside of the cell, and it maintains pressure on the outside of a cell. Plant cells need to have high pressure. It's got a lot to do with water movement, pumping water from underground all the way up through those roots, all the way up of a big, like a 50-meter red gum tree up into the leaves. Okay, they need to maintain high pressure, whereas animal cells don't. All right, so they have that large vacuole. The second thing they have is a chloroplast. Now, I'm sure all of you know that plants photosynthesize, or hope that you do, which means that they make their own food. Okay, they make their own food from light. The organelle that allows them to do that process of photosynthesis is called a chloroplast. Animal cells don't have them. It would be good if we did, because it would mean we could just stand out in the sun and we'd get our nice belly full of food, but we can't because we don't have chloroplast. But plant cells do. We'll have a go at drawing one of those in. So somewhere over here, I'm going to draw. It's a football shape again, very similar to a mitochondria. People get confused between the two, but you shouldn't. There's quite a distinctive difference. If we go back up to our animal cell, mitochondria looked like this. Okay, so it had that double membrane where it had the outer football shape and then that inner curviness to it. Chloroplasts have a outer football shape as well, and they're going to have an inner membrane, but these are like flattened discs. Okay, so I'm going to draw them in with a highlighter here. But they have these flattened rectangular blobs to them. Okay, like that. So that would be our chloroplast there. You can see it looks quite different from mitochondria. Mitochondria's got those nice curved lines. Chloroplasts have those flattened dark discs. Both are football shapes though, generally. Okay, so let's just make sure you've drawn that in and then we'll label it. There's our chloroplast. Now the last thing that plant cells have that animal cells don't is a cell wall. Now a cell wall is like a wall in your house. I said right back at the start that a cell membrane is more like a chuck's cloth or floppy and not like a wall of your house or your room that you might be in now, but a cell wall is. It supports and protects the cell, it provides it with structure. So we're going to have a go at drawing one in again. So get yourself a colour pen and then we're going to draw a second line around our plant cell here, and this is representing our cell wall. So plant cells have a double lining, I suppose, around each cell, one being a cell membrane, which is the same as an animal cell membrane. It allows things to move in and out, similar to a chuck's cloth, but they also have this outer cell wall, which we'll talk about the structure of a little bit later in the course, which provides structure. Okay, it allows for the cell to maintain that high pressure from the water from the vacuole. Now, a quick note here, I suppose, which I didn't mention before, cell walls are found in plant cells, but also in bacteria. Okay, so cell walls are not only found in plant cells, but they are also found, if we go back up the top, in prokaryotic cells. So you can see with this drawing I drew, it had a double lining around it here. Okay, and that's representing a cell wall as well. So one similarity there between prokaryotic cells and plant cells. So in other words, animal cells are the only ones that don't contain a cell wall. That's pretty much it for what you need to know for today in terms of structure and function of cells. Just in case my drawings weren't good enough for you, I can't imagine why they wouldn't be. I've provided you with some nice artistic ones down here, which you can pause the video and have a look at if you want a sort of better indication of what a plant and animal cell look like. But like I said, make sure you know cell theory, the definition of cell, you know the difference between plant and animal cells, you know the difference between prokaryotic and eukaryotic cells, and I expect you to know all of those organelles and their structure and function. We're going to have a bit of a test on it next lesson, so make sure you come prepared. Thank you.